Welcome to Masters of Regeneration Radio. Conversations with humans who dare to reimagine our place on planet Earth. Earth is changing fast. Evolution happening in real time. An intimate, circular understanding of nature and living systems. We are the land. The land is us.
Hi everyone, you're listening to Masters of Regeneration Radio, conversations with humans who dare to reimagine our relationship to planet Earth. What is our purpose? It's to help us all reconnect with an evolutionary understanding of life, with a cyclical understanding of nature and its staggering ability for self-organization and regeneration. How do we achieve this? By putting our attention and effort not on criticizing existing models that no longer work for the planet and life as a whole, but by interviewing the people around the world, the heroes who dare to put their attention and focus on creating new models, new relationships with living systems that simply make the existing ones obsolete. Our mission at the Masters of Regeneration podcast is to distribute knowledge to the masses for a practical understanding on abundant human living with planet Earth. Welcome to episode 10 of Masters of Regeneration Radio. Today's guest is Samantha Esselmann. She's a content and curation scientist at the genotyping company 23andme, where she works on the Ancestry team. We're going to talk genetic composition, ancestry, and how sometimes when you want to find someone, instead of going through Google searches or Facebook, you can simply take a look into the oldest text on Earth, your DNA. That's how I found my long-lost second and third cousins through 23andme and visited them in Santa Barbara, California. Samantha joined 23andme late in 2017. As a content scientist on the team, Samantha works closely with 23andme's population geneticists and content writers to craft an engaging and educational product experience. Samantha became passionate about science communication during her Neuroscience PhD at the University of California, San Francisco, where she also captained the UCSF Science Policy Group. In 2016, she served as the BRCA Foundation's Multimedia Science Communications Coordinator, where she produced a podcast series highlighting a diversity of experiences related to hereditary forms of cancer. Hi, everyone. You're listening to Masters of Regeneration Radio. Welcome to episode 10. Today's guest is Samantha Esselmann. She's a content and curation scientist at genotyping company 23andme, where she works on the Ancestry team. In her former life, she was a PhD student in neuroscience at UCSF. Is that correct, Samantha?
Yes.
Yay. Excited to have you here. Welcome.
Excited to be here.
Sweet. So 23andme, for people who don't know what 23andme is, it's a genotyping company. What does a genotyping company mean?
Yeah, so genotyping is sort of differentiating it from another kind of service, which would be something like whole genome sequencing. And so sequencing versus genotyping is really just the method used to get some of this information to generate things like a ancestry composition report. With whole genome sequencing, the technology is looking at essentially every single DNA base pair, but with genotyping, which is what 23andme and most of the other companies out there are doing right now, is just looking at around 600 to 700,000 spots throughout your genome that are particularly informative for these reports that we're generating. So informative about traits or ancestry composition or health in the case of 23andme.
Very cool. So I did, like I was saying earlier, I did 23andme about 3-4 years ago, and it was really interesting because I was born in Colombia. My paternal grandparents were from Lebanon, and I thought, but I grew up in France, and my mom's French and Colombian, and I thought, well,
it would be cool to just see what percentage of each genetically do I have. So I did the test, and it turned out that I'm like 57% European, genetically speaking. But out of the 57%, I'm mostly Italian. I'm like, how does that work?
You're so
like
cosmopolitan.
Southern Italy or something like that. So that was interesting. It was interesting to see that I also had some percentages of Northern Africa. I guess that's the Lebanese part. But I also had some percentages of Sub-Saharan Africa and Ashkenazi Jewish, and very, very few Neanderthal percentage.
Oh, how many Neanderthal variants did you have?
I don't remember, but it was like, below 1%. It was very little.
Oh, wow.
Yeah. So that was interesting. But then I went into the more details of the test results on the 23andMe app, and I saw that I started seeing the differences in story between the haplogroups, and we'll go into that in a second. I'll ask you to, tell us a little more about haplogroups. But on my dad's side, I'm in a very rare haplogroup. There's like one for 23andMe anyway. I'm like 1 in 88,000 people, turns out to be from that haplogroup on the paternal side.
Yeah, it's pretty rare.
But on my mom's side, it's 1 in 54 people. And it's the A2 haplogroup, which is really beautiful because it's kind of like it has something to do with the coastal highway and the migrations of people from Asia into the Americas. So anyway, I'll shut up there, and could you tell us a little bit more about haplogroups and what they are?
Definitely. Haplogroups is certainly a concept that, even the word is a little intimidating. It's kind of a science jargon word, but at 23andMe, we are excited about using a word like that because there really isn't a more accurate word to use in place of it. And it's sort of we're hoping that by sharing a word like that, everyone gets excited about it and tries to understand what it means. But it does sound kind of like a word that you'd make up to win a Scrabble game. It's a haplogroup is, without taking a semester on genetics, it's an assignment or a grouping of kind of similar sets of patterns in people's DNA. It's kind of like if you're grouping people based on something like their zip codes, or maybe because you inherit your DNA, maybe it's like grouping people based on their parent zip codes or something like that. Okay. But you can then use these patterns in people's DNA to trace back many generations, in some cases, thousands of generations. And it sort of helps us build an ancient giant family tree of all humans. And then so with something like 23andMe, you can see where you fit in that family tree of all humans. Like with the letter, like for example, A2, your maternal haplogroup, that's one of many, but it's informative about kind of where that specific part of your ancestry fits in this family tree. And by specific part of your ancestry, what I mean by that is that we're not looking at all your DNA to generate this haplogroup report. It's only looking at specific parts or specific pieces of your DNA. And for the most part, most of your DNA, like most of your chromosomes, there's this random shuffling that happens between every generation. So you get you kind of inherit a random mixture from your parents of what was essentially your grandparents' DNA. And it makes it difficult to then figure out where each little piece of this DNA comes from. Did you get this little piece from your maternal or your paternal grandparent, like your maternal or your paternal grandparents? But there are two small pieces of DNA that don't go through the shuffling in every generation, and it makes the inheritance pattern really simple, really easy to trace back to very specific lineages. And that's actually, if you think of the word haplogroup, I've already talked about a grouping or an assignment of variation in your DNA, but that's where the group comes from. But then the haplo, that comes from the Greek word aplos, which means something that's simple or single. And so one way to kind of remember that is that it is this simple inheritance pattern, and it's coming from a single parent, so from mom or dad. And There's two different flavors of haplogroups, which you're already going to be familiar with. One flavor is traced through the Y chromosome, which gets passed exclusively from fathers to sons. And these are called paternal haplogroups. And the second flavor is related to this special little loop of DNA that everyone inherits from their mothers. It's called mitochondrial DNA. And this piece of DNA is found in the cell's mitochondria, which, as many people remember from their biology classes, is the powerhouse of the cell. And so for these two pieces of DNA, the Y chromosome and mitochondrial DNA, like I said, they don't get shuffled in every generation. So I guess the question then that people have is why don't, if they don't get shuffled and mixed up, how come we all don't have the exact same mitochondrial DNA, the exact same Y chromosome, which would give us the same haplogroups? And the reason there is because for the hundreds of thousands of years of human evolution, the occasional random change pops up in our DNA and gets passed on, which creates new haplogroups. So that's, I guess, the starter version.
That's
might have been a lot. It's hard to distill it down into like one sentence.
No, it's perfect. I don't know. I'm not sure that people remember their biology class. Can we go a bit more into what mitochondria are and what is the difference between why do mothers pass on mitochondrial DNA?
Mm-hmm. Yeah, I think it's a super fascinating question. When I have this molecular biology and neuroscience background, but when I came here and I started learning a little bit more about kind of population genetics and inheritance patterns, this is a question that I found so intriguing, which is why mothers are the only ones that can pass on their mitochondrial DNA. So everyone, men and women, inherit this mitochondrial DNA from mom, but then only mothers pass it on to the next generation. And the science there is actually not quite settled. Mm-hmm. Some theories say that the sperm has, during fertilization, sperm has mitochondria kind of in the tail region. And so maybe it just doesn't get into the egg, the part with the mitochondria. And that's maybe one reason that fathers don't pass on their mitochondrial DNA. Other reasons are basically just if you think of like getting lottery tickets, like the egg has, I think, over 100,000 mitochondria inside. So hundreds of thousands of copies of mitochondrial DNA. And the sperm cell might only have on the order of like 50 to 100 mitochondria inside of it. So that scale might be the reason that you don't see.
That's a huge difference, yeah.
Yeah.
Okay.
Eggs have some of the most mitochondrial DNA of any human cell type. I think there's maybe a couple exceptions to that, but it's just like a huge number of mitochondria. And so it could just be drowning out the mitochondria found in the sperm. There's other theories that I find particularly tantalizing. So One of them is that mitochondrial DNA is kind of labeled by the sperm cell as being like, once the sperm matures into its kind of final form where it goes off to fertilize an egg, there's a theory that it might start tagging its mitochondrial DNA as sort of like, we're tagging it for trash pickup. Like it's ready to go. We're done with it. We don't need it anymore. And then there's kind of a couple ways that can go. So either the sperm itself targets that DNA for destruction and destroys it itself. And so you essentially have sperm cells going toward eggs with kind of ghost mitochondria, where they're just like the shells without the DNA inside. So they can still crank out energy, but they're not able to reproduce themselves. And then the other possibility is that the egg, and I find this one the best, is that the eggs themselves have some way of recognizing their own mitochondrial DNA versus the sperm's mitochondrial DNA. And then they basically kill the sperm's mitochondria. So no one's really quite sure which one of these possibilities is what's happening, but some combination of those.
That's fascinating.
Possibly. Yeah.
I wonder, and so is there just a fundamental difference between the information contained in mitochondrial DNA and the male DNA?
And like the Y chromosome?
The Y chromosome?
Fundamentally, it all boils down to A's, G's, C's, and T's. The code of life, I guess, is what it's called. But I think what is also fascinating about this is that human chromosomes are linear, meaning there's two ends to each chromosome, and the same is true for the Y chromosome. But mitochondrial DNA doesn't have two ends; it's a loop. And in the kind of great family of living things, the creatures that have circular DNA are typically something like bacteria. So this has led people to explore the possibility, which now looks like it's probably what happened, that billions of years ago, so I guess something like, I can't remember if it's 2 billion or just a very, very long time ago, kind of in the ancient past, some kind of cell that would go on to become eukaryotes, or these kind of more complex life with organelles inside. So some proto cell ate or engulfed another cell, which was a bacterium. And so what happens there is that most of the time it just eats it, and that's the end of the story. But at somewhere in our deep evolutionary past, there was a bacterial cell that was eaten that was not that was somehow beneficial to the host cell.
Okay.
And so that symbiotic relationship led to many billions of years later, it led to mitochondria. So it's pretty clear now that mitochondria used to be bacteria that was eaten by another cell. And so in that sense, it is fundamentally different, but mitochondrial DNA carries some really important genetic material for maintaining mitochondria, especially. Although many of the genes that are thought to have once been in mitochondrial DNA have since been kind of transported over or taken over by our other chromosomes. And then almost as the reverse of that, the converse of that is this Y chromosome. That the evolutionary story of the Y chromosome is also incredibly fascinating because it is one of the smallest chromosomes, and it's a sex chromosome like the X chromosome, which distinguishes it from the other chromosomes, which are called autosomes. And the Y chromosome is I guess you could call it the sex determining chromosome because it determines male sex. I guess you could say the default would be female unless you have this Y chromosome present. And the reason that it will kind of, it determines male sex is that it carries this one really critical gene called SRY, which is not a very creative name for what is essentially the sex determining region on the Y chromosome, SRY.
Okay.
And this gene basically starts this cascade of inside the developing embryo of kind of putting the developing embryo on the path toward becoming a male. So there's so many things to talk about. This is an outlet for me to talk about all the stuff that I think is cool.
No, it's fascinating.
So the Y chromosome, there's evidence that maybe many generations ago, and we're talking again millions of years, there's a pretty good chance that the Y chromosome used to be the partner for the X chromosome. So it used to be the exact same chromosome. But then sometime in evolution, it got this SRY gene that determines male sex, and at some point, it didn't really need all of the other stuff that's on the what is now the remaining X chromosome. And so it was just starting to lose gene after gene after gene until one of the only really important genes left on the Y chromosome is this SRY gene, and it's much smaller than the X chromosome, which led people to think that, maybe the Y chromosome is going to disappear completely. But then since then, scientists have sort of figured out that no, it looks like it's actually pretty stable, and this is a pretty good system that it's figured out where it just needs to really be holding this SRY gene. So That's an interesting story too. But they're both important, obviously, the Y chromosome and the mitochondrial DNA.
I'm fascinated with how information gets passed on from one place to another. Biological information. I'm not talking about emails.
Yeah.
Right.
That's fascinating too, but
That's fascinating
not
too,
something I
but-
can talk about.
No.
Yeah,
How
I
does
think.
that shuffling happen?
Oh, sorry. Go ahead.
Well, no, just kind of thinking about how then those two come together and how they communicate and how they get shuffled from one generation to the next is.
Yeah, so recombination is that shuffling, kind of biological term for that shuffling that happens. And it is in human evolution and for most living organisms, recombination is critical for kind of preserving genetic diversity in the sense that if you never recombined, if there's like a mutation that is not beneficial, or if there's something like a disease sweeps through a community and they all have the exact same genetic background, maybe that whole community gets wiped out. But if there's some genetic diversity in that community that can be maintained with something like recombination, then there's a slightly better chance that community, even if some people don't survive whatever bad disease comes through, there's a pretty good chance that there's enough coming out the other side of this epidemic that who had this protective genetic background because of recombination, that the species survives. I'm not going to be able to remember all the way back to all my biology classes in undergrad, but recombination. There's even like videos of recombination, or there's videos of cells going through the different cell cycle, the phases of the cell cycle. And so you can see the chromosomes kind of coming together, mixing up, and then pulling apart, and the cell becomes two cells. It's just a really fascinating process. Even getting back to the Y and the X chromosome, even though they're not at all similar to each other anymore, they look very different. The X chromosome is a very large chromosome. The Y chromosome is a very small chromosome. There's still evidence that at the very tips of these chromosomes, they do have some similar material and they can recombine at the very, very tips. So that's kind of a hint about their ancient evolutionary relationship.
And so, yeah, I just had a thought, like, life does that with every single organism on the planet, arguably, to look for diversity and promote, I don't know, so that diversity which gives you resistance to whatever shows up and that's going to attack you and affect your life. And how we can relate that to because we talk a lot about food on the podcast and regenerative agriculture and all that, to like how monocultures just create the opposite of that, right?
Yeah.
It's creating less and less and less diversity, and that sounds kind of counterintuitive.
Yeah, because if you think about it, there's an argument that can be made for basically a monoculture or organisms that reproduce asexually or without this sexual recombination. The argument is like, well, you can just make, hundreds of yourself really quick, just boom, boom, boom. You don't have to wait for, a partner to come along, and they're all genetically exactly like you. So you're kind of propagating yourself way more directly. But and this works. There's examples of this working for at least a few generations where one organism can become incredibly successful and take over an ecosystem. But long term, a lot of the organisms that are able to reproduce asexually, some of them also have the ability to sexually reproduce so that it can kind of pick and choose where necessary. I think that's right. I'm pretty sure I just saw something about that, but maybe we don't talk about that too much, because that's kind of where my knowledge starts hitting a cliff. But there's a balance. There's like, sometimes it can be really powerful, but then one bad thing happens and it wipes out the entire species. But if you reproduce asexually, you have more of that diversity. So I think that is a beautiful evolutionary story.
Yeah. That's interesting. We'll have to do another episode on that subject. I remember when I did 10 years ago the ancestor of 23andme,
Yeah.
It came with a really cool documentary that explained how some doctors had started going around the world to actually look for these blood markers that allowed you guys and all the companies doing that to trace the journeys and the migrating patterns of people.
Mhm.
Is that like a global open database that's been created that you
Yeah, so
23andMe can access and other companies as well, or how does that work?
so we actually, there's a couple databases that are kind of these global databases that we can pull from that are available to everyone. And that's, I think back in the day, how I wasn't here then, but that's how we generated our initial kind of reference population. So our continent level reference populations. Let me see if I can find the names of those sources. I think one of them is like the HGDP database. There's like the Thousand Genomes database. Oh yeah, so when we the one of the recent iterations of our reference populations, there were around 11,000 unrelated individuals with known or self-reported ancestry, and of those 11,000, close to 2,000 came from publicly available data sets. One of them is called the Thousand Genomes Project. And then there's also the CEPH-HGDP and HapMap. So I'm not that familiar. I can't speak to what
Cool.
those projects did or how they developed their data sets. But for those 11,000 individuals, some subset comes from these publicly available databases. And now, because we have such a large database ourselves, we're able to generate some of the reference data set ourselves.
Cool. Maybe if that's cool with you guys, maybe later you can send me some links that we can put on the episode so people can just geek out on
Totally.
databases and stuff like that if they're
Totally.
interested.
Yeah.
If anyone. Um,
People are into some weird stuff these days. That's
yeah.
fine.
So here comes the second part of my funny 23andMe story.
Okay.
So my paternal grandparents left Beirut, Lebanon, in the Middle East, back in 1914 or something like that, right?
Yeah.
And my granduncles, so my grandfather's brothers, went to Monte Carlo, Pennsylvania, California, and you know, they
All
settled
over the world.
in other places, right? So they were all in
Mhm.
three different countries. And they had children, and my dad and my aunt met some of those descendants of my granduncles back in the 70s. So that's like almost 50 years ago.
Mhm.
And then they lost touch. There was no email, nothing, right? And then I'm on 23andMe. I opened the app just to check if there's any new details last year. This is like 3 years after I did 23andMe. And I have a message from a guy that says, "Hey, what's your story? I think we're related." And I see and I look and it says second to third cousin. And I'm like, "What?" And so I tell him the story I just told you about my granduncles. And he says, "Yeah, your granduncle Emile went to Monte Carlo and he died this year. Your granduncle
Oh,
Jim went to LA and died this year. And your other and he knew everything. He had done the whole family tree and the whole thing. So it turns out he's about my dad's age. He's 80 or 78 or 80 years old.
mhm.
And
Wow.
he is my grandfather's cousin, son.
Grandfather's cousin's son. Yeah.
Yeah.
Some of these relationships are so hard to describe.
Yeah,
Like
yeah, yeah,
the
yeah. I
name
know.
you put on it, if it's like a second cousin once removed, like yeah,
Yeah.
it's hard
So
to keep track.
I'm like second to third cousin. I don't even know what that means, but it's definitely closer than all the genetic relatives,
Mhm.
that you get on 23andMe that are 5 or 6 degree of separation genetically.
Yeah, the fact that you're actually able to trace that relationship,
Yeah.
that's a good
So we
start.
share
Mhm.
great grandparents at some point or something like that.
Cool.
And so, but the beautiful story around that as well is that he's like, yeah, my dad was very good friends with your granduncle Nicholas, who lived in Santa Barbara, California, and made violins. He made instruments, right?
Wow.
And then I tell his story to my dad. I'm like, oh my God, I found these guys through 23andMe. And he's like, oh my God, I can't believe it. My dad used to tell me stories about my uncle who made violins in Santa
Oh,
Barbara. And I'm like, holy shit, this is awesome. And I finally met them last week. I was in Santa Barbara and we met in person there,
wow.
they're almost in their 80s, and I don't know if I'm going to see them again or not. And
Yeah.
but I found out not only that they exist and they have children, and they're Norwegian, Lebanese, American,
Wow.
right? And they're in Minnesota and Wisconsin, and then, but through them, I found that there's one person that they know of in Lebanon today with my last name. So like an even
Okay.
more closely related parent.
Wow.
That's the short story. And I opened 23andMe again like last week when I'm with them, and we find out that there's another guy who's Brazilian Lebanese with who we share an even higher genetic percentage, like 3% or something like that. So I don't know. It's like the genetic social network or something like that. It's the Facebook of your genes. You know?
Yeah, I mean, that would be like, I don't really know what our high level goals are with that kind of thing, but because I'm focused on the ancestry stuff, but I do know that that would be a really cool place to end up if it became this kind of, genetic social network. Yeah.
Well, it is in a way. I mean, for people who take the 23andMe test anyway.
Mm-hmm. And so, yeah, and some people love using that feature, and they message all their relatives, and they get these great, family reunions, and
Yeah.
they can piece together their whole family tree that way. And then the other people just kind of think it's cool to know that they have all these relatives out there. So,
Yeah. So I did the test for both the Ancestry and Health, and I also, I mean, thank God nothing came out, positive, like weird conditions or something like that. But I also got like exactly how I knew. I mean, I knew intuitively that I was lactose intolerant because I hated milk when I was a kid, and it just like makes me bloated and tired and whatever. And that I also have the genetic variants for celiac disease, but I'm not celiac,
yeah.
But gluten is really hard on my gut. So yeah, it's fascinating. Do you have an interesting story of your doing the test? Anything that
I think so. Nothing really popped out at me. Of course, I did the health plus the ancestry. Unlike your story, my ancestry story is a little bit more, I'll say, blue. So if you look at the map, the European source populations, you'll see that reflected in a variety of blue colors. And so my entire map is blue. All my if you see the chromosome painting on the report where you can see where like the origins of different pieces of your chromosomes, that's all blue. I'm all European.
Yeah.
And my family has actually been in the States on every side for many generations. And so that, I guess, sort of mixes the pot in terms of whether I'm, British and Irish, Scandinavian, French and German. And so in that sense, it's almost it's murkier for me. And so some people are getting these country hits or these regional hits where their recent ancestors come from. I don't know if you've seen any of those results for you, but I only get the United Kingdom and Germany. And then there's some people who are more recently from Europe with a little bit more mixed families, and they have, the UK and countries in the Balkans and Italy. And so you can have very exciting results from Europe, but I think my family is just a little bit too American at this point to have that. And then for my health reports, again, I think I was lucky. I think most of the results were encouraging or not scary. There was one interesting variant that I carry for hereditary hemochromatosis, which is common. It's sometimes called the Celtic Curse. It's very common in the British Isles, but then also a little bit in the kind of just Northwestern Europe in general.
that's a lot of iron in your liver or something like that.
Exactly. It's kind of the opposite of anemia in a sense, where you have very high levels of blood iron, and it only kind of pops up in middle age or later for some people. But I only have one of the variants, so it's not really something I have to worry about, because I would have had to have two of the variants to have been at risk for the disease. But that led me down this whole rabbit hole of figuring out like the kind of geography, the patterns of that variant, that disease around the world. Let's see, what else has been interesting.
So you don't eat a lot of red meat or at all?
Well, so
Or
for me, it's
how does
not a
that work?
problem because this is one of the carrier status
Oh,
reports,
you
and
have
for
the genetic
those,
variant, but
yeah,
it's
I have
not
the
active.
variant. Hmm.
Okay, gotcha.
So I have to have two copies of that variant to have the disease, and I only have one copy. So it's useful information. I have a partner. My husband has taken the 23andMe test, and he does not have that variant. And so I would say there's very little chance if we in the future decide to have children of our kids having hereditary hemochromatosis. So that was an interesting thing to see that pop up, but then to know I don't have it. My mother has also done the test, and my father has done the test, and they both have like one or two of these carrier status report variants that they did not pass on to me. So it's kind of fun to see the like, woo, I got the good copy of that from mom.
Yeah.
So, and then in terms of my like traits, they came out recently with some new kind of small traits reports that are related to like your ability to match a musical pitch. And before I did any science, I was actually a music major. And so that resonated with me because I was like, oh, well, I can match. According to my genetics, I'm predicted to be able to match a musical pitch. And then they also had like a mosquito bite likelihood based on your genetics. And I'm more likely to be bitten by mosquitoes, which matches my experience.
Because the mosquitoes can smell your genes or what? How does that happen?
Oh, I can't actually remember what the science of it is, but yeah, it must
Talk
be
about
there. They must be
biological
smelling
information
something.
flows. Like,
Mm-hmm.
how does a mosquito find out about that?
Yeah, yeah, it's fascinating. There's
So,
also like
okay.
there's also the wake up time report that they came out with recently, which is predicting if you're like a morning person or a night person based on your genetics. And for me, it matches pretty well with what I know about my sleep cycle. For some other people, it doesn't quite match as well, but
Can you read
it's
about the
interesting.
science of all those new test results on
Yeah,
23andMe
I
or
think I'm pretty sure. So I don't really work on the
yeah.
traits side of things, but I'm going to go to my report right now and see what it can tell me about the mosquito bite frequency. For a lot of these reports, we might not even fully or even at all understand the molecular biology of like why this thing is happening or this thing is happening. But if you start at a trait or what's called a phenotype, something that is not necessarily based on your genes, but is like something that's outward facing that you can something that you do or like a hobby that you like, it can be anything. We can
Okay.
take that information from these research surveys and run what's called a GWAS, which is a genome-wide association study, and correlate the different traits with people's genes. And we can find these variants they pop up in this study, and we say, okay, this one's associated with this trait. So say getting bitten by mosquitoes, but we're not going to necessarily from just that study be able to tell people why this is happening. All we can say is that it's an association, which I think is still very interesting. But
Yeah.
let me see if I can go to the
I
trait
just find it
reports.
fascinating to think like when, and here goes another question, but before we jump into that, just like how will we ever be able to measure, because that sounds like there are things, what you're saying is, it sounds like there are things related to culture, like the ability to sing or to do certain things. And but they're reflected in your biology. And when will we be able
Yeah.
to measure kind of like how your biology becomes your culture or your culture becomes your biology? That's just a fascinating subject in and of itself.
Yeah, so that's totally like the nature versus nurture debate. And there's kind of a spectrum where there are some traits that are completely determined by your genetics, and then there's some traits that are almost completely determined by the environment or by
Yeah.
nurture.
And then there's
So
so 23andMe measures all of these traits that define us. And then there's all are you guys doing anything with epigenetics? Or is there a way to measure that? Is there a way to measure how gene expression, manifests?
yeah, so I'll definitely say there are ways to measure that, and I think there might
Okay.
even be companies out there that look into that, but we are not that's not something that we currently offer at 23andMe, and I can't speak to what the research team is trying to develop, mostly just because I have no idea. But that is a whole other world of information that probably has a lot to do with your traits and how that's kind of really, I think, closer on the nurture side of the spectrum. It all comes together in such a complicated way. And I'm not sure that, in 10, 20, 50 years from now, that we're really going to have a handle on how that all goes together. Female mosquitoes have a complex olfactory system that lets them sniff out their food. As it turns out, mosquitoes have preferences. Mosquitoes are attracted to certain molecules in body odor and breath, and depending on the proportions of these molecules, some people may appear more delicious than others. So I guess that's a simple answer. And then if you go to the report, there's there's something like 10 links to primary literature that can talk about some of the more molecular aspects of that. But our report is mostly just picking up the associated variants that may or may not have any, causal link. That's the nature of these reports, is that's something that's hard to know.
How do they perceive that? How does a mosquito perceive that you're more delicious? How do they understand those molecules? It's just, how do they get those signals? Fascinating.
That's probably someone can spend 7 years in a graduate degree trying to figure that out. So
totally.
It's a lot to unpack.
Yeah. It's like so many, if the oldest language on Earth is the genetic code, there's still so many myriad languages within that language, and how it flows from one organism to the next.
That's a great way to say it. Yeah.
Yeah, it's fascinating. Yeah.
So I can also just say specifically for the mosquito bite report, if it's kind of helpful to understand what that might mean. The basic report tells me that I'm more likely to get bitten, but then there's a breakdown where it shows that with people who have very similar genetics to myself, who have very similar variants that are associated with this report, the majority, or 55% of the people with similar genetics, report more mosquitoes bite them than bite other people. But there are still around, 29% that report about the same number of mosquito bites, and about 16% that report fewer mosquito bites. So there's still this spectrum where you can have the exact same genetic breakdown as myself, but you might still be reporting fewer mosquito bites than others. So, it's not a...
Does yours say that you're likely bitten more often than others?
My report does say, Samantha, based on your genetics and other factors, you are likely to get more mosquito bites than others.
Me too.
Oh, congratulations. Aren't we lucky?
That's ridiculous. And it's fun. Cool. Is there anything else that we've left out that would be interesting to talk about?
There's so many things. I think I before I worked here, I thought, diversity is beautiful in this kind of abstract way, and human migration, like the story of human migration, is beautiful. But working here has given me a chance to really kind of pick at all these stories from all around the world. And I think something that I've found kind of intriguing is that, the archaeological record of human history and human migration has been very informative and has told kind of one side of the story. And you can pick out, like, this skeleton here looks kind of like this modern population, or this skeleton over here is, more related to this population. But the genetic record that we're uncovering in living populations today, like, you can say, how does looking at the genetics of existing populations, how can that tell you anything about human history or what came before us? And that record is actually incredibly informative to kind of look at the patterns that exist around the world and extrapolate from that, like, where this group likely migrated, or this group, based on their genetics, likely mixed with a group that shared similar ancestry with another group over here. And so you can kind of fill in all the gaps of the archaeological record. Another interesting twist on that is not just the archaeological record, but the kind of written record of human history, you often hear this trope that like history is written by the victors. And so you can say something like in the British Isles, for example, about 2,000 years ago, the Romans invaded, and then the Anglo-Saxons came, and then the Vikings arrived, and then about 1,000 years ago, the Normans came from France. And we might think that because these groups all ruled England at various times or ruled Great Britain at various times, that there might be a big record of that in the DNA of modern British people. But if you look at their DNA, some of these groups actually had a much larger genetic influence than other groups. So, for example, even though the Romans technically controlled Britain for, I think, something like 400 years, there's actually very little evidence in the genetics of modern Britons that they had this kind of Roman period in their history. But then there's actually a very, very big imprint from this Anglo-Saxon invasion. And so that kind of tells you about the relative scale of like, even though the Romans controlled this place for hundreds of years, like it must mean that it was just kind of this ruling class, or maybe the Romans who ended up in Great Britain were not the Romans from Rome. They could have been other Romans from the wide Roman Empire, like from northern France, who are actually genetically very similar to the people of England anyway. So I think that's kind of interesting how, genetics tells a different story than what's written in the history books.
Oh, totally. It just breaks through the history books, like you say, or through the belief systems, that someone might think that they're so pure racially or genetically, and turns out they're a big mutt like me, for example. Or,
Oh, and that's the other like totally fascinating thing to me is I knew that humans evolved within Africa and then migrated out of Africa and kind of spread all over the world. But what was not necessarily clear to me or intuitive to me before working here is that pretty much all of the human genetic diversity that we see around the world, and so I'm not talking about like physical diversity or phenotypic diversity, but just like what's in your genes. Like at this position, there's a variety of base pairs or of DNA bases. That genetic variety are found around the world can all be found, or can almost all of it can be found within the continent of Africa. And that represents sort of this kind of series of migrations outside of Africa and beyond, but it all comes back to Africa, and most of that genetic diversity can still be found within Africa. Whereas if you take someone from, England and someone from Japan, they're probably going to be genetically more similar to each other than if you take two people from somewhere in sub-Saharan Africa.
Wow, that's amazing. So still today you would do this, sequence someone's genes from Africa, and genetically they'll be more diverse, or they'll have that. Wow, that's awesome.
And so it's so hard, I think, without that context to understand that just by looking at someone, you say, I can see that their skin is a similar color, and I can see that their features superficially seem similar to me. And so I'm going to assume that they're genetically similar, but that's not at all the case. And a lot of genetic variability has nothing to do with the physical features that you see, like in someone's face or their hair or their skin color. A lot of that variability goes into lots of other complicated things about our biology, so that to people who might, especially to someone from Northwestern Europe, if you're looking at people from sub-Saharan Africa, like historically, we have kind of lumped sub-Saharan Africa into this one box. And it turns out that is like the most genetically diverse place on Earth.
How do you measure genetic diversity?
Pretty much in the exact same. So in genotyping, we look at SNPs, or these single nucleotide polymorphisms. These variants found. I just threw that at you. So the variants that I've been mentioning, that's just like if you look throughout the genome at the different DNA bases, you might have a stretch that says A, T, C, G. The SNPs are in most humans, on average, most humans are going to have that same sequence, A, T, C, G. But at the second site, at the second A, some percentage of people around the world have a T there. So instead of AATCG, it would be ATTCG. And that switch from an A to a T, that would be a variant or a SNP, a single nucleotide. That's the A or G or T. Polymorphism, which just means difference or change. Okay. So we measure SNPs, and I think humans have like a total of something like 2 million. I might want to check that number, but something around like 2 million sites where these changes appear throughout the entire human population. We're the exact same at the other, pretty much 3 billion base pairs. But at these 2 million or so sites, a very small fraction of that 3 billion base pairs in our genome, there are differences. And so those differences are kind of what we're looking at in our test. And most of those SNPs or most of those differences are found within the continent of Africa. But if you go somewhere like Norway, they're not going to have quite the same breadth or depth of diversity of those variations or SNPs. I hope that made sense.
Yeah, totally. I'm just wondering, like thinking about migrating patterns and evolutionary just processes and time. Mm-hmm. How was Africa so diverse genetically?
Yeah. So one way to think about it is humans actually are genetically not diverse in the scale of other species. So like the common chimpanzee in, I think, West Africa is like in the area of like Cameroon and Nigeria right now, the common chimpanzee has so much more genetic variability than the entire species of human. And this is just the chimpanzees that live in like Western sub-Saharan Africa. And it kind of reflects the fact that our species is a kind of young species. And that the genetic term for why some of this might be happening is called a genetic bottleneck. And that's actually, if you think about the image of a bottleneck and stuff passing through that bottleneck, it kind of funnels things down, and all the stuff that's inside the bottle might not be able to make it out the other side of the bottle. So it's commonly illustrated with a bunch of brightly colored marbles inside a bottle, and someone's pouring it out, and what comes out the other side is only a subset of the brightly colored marbles that are inside the bottle. And so if you think about human migration and passing these kind of spreading or pouring out these variants into the rest of the world, when humans migrated out of Africa, the vast majority of the human species stayed within Africa, and only a few thousand or so left by way of, the Sinai Peninsula or crossing the Red Sea or however they did it. And that small group was carrying only a subset of all of the diversity that existed in the places they were leaving behind in Africa. And those groups then went on to South Asia, East Asia, Oceania, the Americas, Europe, and their starting material was more limited than the starting material found within Africa. So I have to log back into my computer. I went to sleep. I was talking for so long. So this bottleneck phenomenon, there's lots of reasons a genetic bottleneck can happen, whether it's kind of a founder effect, which is when a small group, sails off into the unknown and colonizes a remote desert island. There's only, again, you're going through this bottleneck where you have a subset of the genetic variation that from the population that you originated from. Other examples in human history, apart from the kind of main out of Africa migration, something like the founding of the islands in the Pacific Ocean, or the people who came across to the Americas around 15,000 years ago, like that migration. There's, if you're thinking about the haplogroups that we talked about earlier, there's essentially one primary paternal haplogroup based on the Y chromosome that went on to found all of the kind of indigenous people of the Americas. So that means that like one or very few men sort of led to the entire population of the Americas. And that's not to say that that's strictly what's happening, because there could be more men, but just the Y chromosome variation that we see today in the indigenous peoples of the Americas is much less diverse than the Y chromosome variation that you'll see in Africa.
Gotcha.
Sorry, I'm dumping a lot.
No, it's so good. It's fascinating. And can you measure, I don't know if this makes any sense, but can you measure what genetically, how much of you is made of like non-human genes, like, I don't know, plant or other animal?
I mean, there's very simple metrics. Like you hear these stats all the time that like you are, I don't know what the exact number is, but like you share 50% of your DNA with a banana, you know?
Yeah, things like that.
Yeah, there are pretty basic ways to measure that, because there are genes that code for proteins that are actually shared across much of life. And obviously there's some organisms like chimpanzees that are much more similar to humans than a banana would be. So it is measurable. I'm not exactly clear on how that's calculated, because there's all sorts of things that go into that. Like, are you talking about, the general protein sequence is the same? Or are you looking at all the non-coding kind of like dark matter, quote unquote, dark matter DNA that exists? Like, I think that's a little unclear to me how that's calculated.
Dark matter DNA.
I think people who study this DNA would be upset that I'm calling it dark matter DNA. I think that's just, people don't know what it does, but that doesn't mean that it's pointless.
Like your virome or something like that? Or, I don't know.
Yeah. So there's all the other, so many things there, like all the non-human things that make humans work, like that over half of the cells in our body are non-human.
Yeah.
They're bacteria. For the most part,
etc.
Yeah,
that would be a super fun feature, though. If we could compare, like, oh my god, like how much banana are you, and you know.
Oh, it's been talked about, I'm sure. I think we send out those little factoids every once in a while on like Twitter. We'll say we're 50% banana. I think it's hard for people to conceptualize what that actually means for them, but
I think it sounds funny, but it would be such an amazing insight, because most of us, we think of ourselves as separated from nature. We're like, oh, we live in cities, we're humans, you know.
Yeah.
Or we're just made of the same stuff. That's just transferred. So it would be fascinating to see, like, at an individual level, like, oh my god, you know, in the great shuffle of evolution.
Yeah, the banana is your very distant cousin.
Yeah.
Totally.
Anyway, Samantha, thank you so much for the awesome, enlightening interview.
Sure.
Such great information.
Love talking about this stuff.
Yeah.